For those of you at home watching this, all three of you, maybe that's generous. We are going to spend the first few minutes reviewing over DNA structure and replication. If you need a detailed explanation of that, I have posted it under days 1 and 2. And then in about 10 or 15 minutes, we'll get into the new stuff for today on protein synthesis if you need to fast forward ahead. So, few things you need to know. The test on genetics is planned for February the 10th, which is a ways away, but that should let you know that there is a lot of stuff on the genetics unit. There's actually really 2 units here that make up genetics. One's on molecular genetics, which is the small scale, how DNA works, the molecular level, and then there's Mendelian genetics, which is like Punnett squares and pedigrees, how traits are passed from parents to offspring. And that's what we're going to be working on for a little over the next month. You can imagine there's going to be a very large test at the end of all this. This is by far the most complicated unit that we will have all year. So you guys really need to be on your game this semester. Ecology, evolution, and genetics, which are this semester, will make up probably 60% of your AP exam, if I had to guess. So genetics is probably the largest section out of the exam. So you guys really need to understand what's going on and have a good, you know what's happening with these different concepts. So on 317 at the bottom of the page, DNA, if DNA, so you've got deoxyribonucleic acid, you've got ribonucleic acid. So nucleic acids make up one of the 4 types of what? Nucleic acids are one of the 4 types of, see, we've done some mind-altering drugs over the break. One of the 4 types of macromolecules. You have nucleic acids, you have proteins, you have carbohydrates, and you have lipids. So DNA and RNA are nucleic acids, one of those types of 4 types of macromolecules that make up living things. Now, DNA is a macromolecule, so it's made of a polymer. It's made up of many monomers. So if DNA is as a train, what are the cars on the train that make it up? Do you might know the monomers of DNA? I mean, what'd y'all do yesterday? This was the first thing on yesterday's notes. The monomers of nucleic acids are nucleotides. Good. So if you're wondering at home, is anyone in here? Yes, but it's only 3 people and none of them have any idea what's going on. So nucleotides are the cars on a train or the monomers that make up DNA. What are the 3 components of a nucleotide? Who knows?
A base, a sugar, and
Nitrogen base, sugar, and phosphate. Good. So a nucleotide is made up of those 3 components. On 317, you can see here, if DNA is a ladder, like you see on the board here, the outside part of that ladder of DNA is made up of what 2 things? What's the outside part of DNA made of? The sugar and the phosphate. Are y'all looking on 317 there? There's a picture of this in the middle of the page. So on 317, you see the big blue thing that's like a pentagon? That is the sugar. In this case, it'd be deoxyribose. In RNA, it would be ribose. Okay? And the little circle thing with a P in the middle of it, that is your phosphate group. So basically, all up and down the steps of the DNA ladder on the outside, you have sugar, phosphate, sugar, phosphate, sugar, phosphate, okay? In the middle, the steps in the ladder are the different what? Nitrogen bases. So these are your A, T, C, and G, your adenine, thymine, guanine, cytosine. And in the case of RNA, this would include Uracil, good. So remember, there is no thymine in RNA. It is replaced with uracil, okay? So now, some other things to take note of here on 317. DNA runs in opposite directions, almost like a two-lane road. One goes one direction, one goes the other direction. And you'll notice this on the page, it says 5 and it has an apostrophe. They call that 5 prime if you watch a review video. And at the bottom, it's got 3 prime. And the opposite is true on the other side. So they're running in opposite directions. One of the most important things to note from part 2 of your notes is that DNA and RNA always are copied and read in the 5 to 3 direction. Always. Always copied, always read in the 5 to 3 direction. Okay? So when we are copying DNA and we're copying, making proteins here in a second, toward the end of class, that is always true. Okay? So they're always going, and when they're added on in that 5 to 3 direction. Okay? Now, let's look on page 324 and 325. So on 325, at the top of the page, there's a big chart with lots of proteins and enzymes that help in DNA replication. I would say you need to be familiar with all of these except for the topoisomerase, only because I can't say it. No, all of these show up in your notes except for that topoisomerase. The rest of them are important, and you need to be familiar with them in the process of DNA replication. Now, I guess we'll back this thing up for a second. Why does DNA replicate, Mason?
So the cells can replicate?
Yeah, in order for a cell to divide, you have to have 2 copies of DNA. Good. When does DNA replicate, Katie?
Interphase.
S phase of interphase. Good. So those are important things, the when and why it happens. Where does replication take place, Mariah?
In the nucleus.
More specific.
The membrane.
You were right on the first one, but I want you to be more right. Mason?
The nucleolus?
There you go, the nucleolus. So, when, where, why, we got all the important stuff. 324, let's read through these and make sure we understand the replication process. If you did not do this at home, did anybody watch the video with Bozeman or Crash Course on DNA replication and structure? So, I would say both of those are good videos. If you didn't quite get it down with one, maybe watch both of them. Bozeman Science and then the Crash Course videos on DNA structure and replication are really good. This is a very complicated process. There's a ton of enzymes that you need to be familiar with. And just understanding how the leading and lagging strand works is pretty complex in and of itself. So I was really disappointed, I'll be honest with you guys, from my first period and when they came in today, and it didn't seem like they hardly did anything on the break. I'm not sure about this bunch if they did anything or they're just afraid to answer, but you guys are going to have to bring your A game this semester if you are going to do well on the AP exam. And I fully think you're all capable, but we are short a period and we have a lot of information to go over and we're already a little bit behind. So you guys need to make sure that I try not to give you a lot of homework, but you're doing what you need to at home to be successful. So anyway, number 1, Katie, start us on 324 there.
Helicase unwinds the parental double helix.
All right, DNA is in a double helix ladder shape where it's twisted. And in order for replication to take place, the DNA has to unzip or unwind. And the enzyme that does this is called helicase. Good. Number 2, Mason.
Molecules of single-strand binding proteins stabilize the unwound template strands.
So I like to think of the hydrogen bonds that are holding the nitrogen bases together in the middle. I like to think of them almost as like magnets. They're not a strong bond, but they still hold it together. And when you pull them apart, they have this tendency to want to come back together. So you need something, once helicase unzips that DNA, to hold it apart. And so what holds it apart? You literally just read it, Mason.
There are molecules of single-strand.
Single-strand binding protein, good. So these single-strand binding proteins help to hold that DNA apart so it doesn't snap back together. Number 3, Mariah.
The leading strand is synthesized continuously in the 5 to 3 direction by DNA pull 3.
DNA pull 3 is short for DNA polymerase 3. Polymerase is an enzyme that's making making a polymer of DNA. And we're going to talk about DNA polymerase 3, which adds in nucleotides continuously to the leading strand. And we're also going to talk about DNA polymerase 1. I don't know what happened to DNA polymerase 2. They don't mention it in your book, so I don't really care. But on the leading, so this is probably the most confusing part of replication. There is a leading and a lagging strand. So these replicate a little bit different. What they always do, DNA always replicates in what direction? 5 to 3. So as the DNA unzips, the leading strand that can continuously add in nucleotides in the 5 to 3 direction. You see how it's opening up a new section there as it unzips, and it can just continually add in that 5 to 3 direction. Now, really quick, just so that you have an idea of why this is. 5 and the 5' and the 3', I hope you don't have the fortunate or unfortunate task of taking organic chemistry in college. But if you do, you'll be involved in naming compounds and how carbon compounds are named and how they work and a bunch of other stuff that, I don't know, my brain exploded. So the 5' and the 3' are just the air. You remember the sugar, how it had that 5-carbon sugar, deoxyribose? So the 5 and the 3 are just like the locations, like the 5th carbon and the 3rd carbon where stuff is attached to. So that's all the 5 and 3 mean. So it's got to always attach on that 3 end. So it's going from the 5 to 3 direction. Okay? That's what the 5 and the 3 mean. And really you don't need to know much about that. I just didn't want you to wonder what the heck that came from. So anyway, now let me back up a little bit here. DNA, look on the bottom, look on the top of 321. DNA is really long. You have about 6 billion nucleotides of DNA that have to be copied in a few short hours. You have 23 different pairs of chromosomes. Each pair of those chromosomes have millions of nucleotides on them. So you could unzip it from one end to the other and copy from one end to the other, but that would do what? Take a long time. So in order to hasten this process, what happens is at multiple points along that DNA strand, it unzips. There's a little code there. It unzips, and it starts replicating in both directions. And at the top right of 321, you can see here it creates these replication bubbles, and it works out in both directions. And in doing so, it speeds up that process. So you've created these bubbles where it's replicating at multiple places on the chromosome, and they're both working in both directions instead of starting at one end and just working your way to the other. In doing so, it quickens it up, okay? So I wanted you to get your head around that. So on this overview you see on 324 here in beige, you can see there's an origin of replication where it starts, and then it's working out in both directions where the DNA is unwinding, unzipping, and new nucleotides are being added into each side to help make 2 new strands of DNA, okay? So the leading strand, as it unzips, can continually add nucleotides in the 5 to 3 direction as it unzips. The problem is, as it unzips on the other side, 5 to 3 goes from, in this case in your book here, 5 to 3 goes from left to right. So can it continuously add in nucleotides as it unzips? No, because it can only add 5 to 3. So it has to wait for it to unzip, and then it will start to add in nucleotides back in the other direction. And once it gets to where it started, then it has to wait for it to unzip some more and add in some more nucleotides. And what you wind up with, if you look here at the overview, you wind up with a bunch of little fragmented segments of DNA that's added in, okay? And then those have to be all hooked together and made nice and neat in order for this DNA to work. So, how does the lagging strand work? Number 4, Katie.
Primase begins synthesis of the RNA primer for the 5th Okazaki fragment.
Okay, so these little fragments of DNA that are made in this process as it waits for it to unzip and then it copies backwards, these little fragments are called Okazaki fragments. They're named after a Japanese couple that figured this out. More power to them. And that's where that term comes in. But a couple things you guys need to know. Every time that DNA is started to, that you're going to start to replicate DNA at the starting point, you see at the origin there, each time a new segment is made, RNA, a little piece of RNA has to be laid down, a primer, okay? Now, that's done by the enzyme primase. Now, so if you think about it in this context, like when you paint something, most people normally use a primer. They prime it to cover up colors and stuff, but they also give it something like on cars, that primer gives the paint something to stick to. And so in this case, you can think of that little RNA primer as something for the DNA to stick to and hook onto and start replicating. So a little piece of RNA is laid down by that enzyme primase. And then, number 5, Mason.
DNA polymerase 3 is completing synthesis of fragment 4 when it reaches the RNA primer on fragment 3. It will detach and begin adding DNA nucleotides to the 3 end of the fragment 5 primer in the replication fork.
Yeah, that sounds confusing. Basically what happens here is you have the RNA primer that's laid down by primase, and then this enzyme, the same one used in the leading strand, DNA polymerase 3, then it attaches there and it starts laying down nucleotides from that 5 to 3 direction until it meets the previous fragment. Okay? So it's doing the same thing it did in the top, it just can't go as far because it's waited for a little bit of the DNA to unzip, and then it's having to go backwards in this 5 to 3 direction until it meets that previous Okazaki fragment. Now, what can you not have in your DNA?
Mutations.
RNA, good. So, yeah, we hope they don't have mutations either, Mason. So, we've got RNA though, right? Because we laid down an RNA primer. So what do we need to do? Remove it. All right, number 6, Mariah.
DNA polymerase 1 removes the primer from the 5 end of fragment 2.
Okay, blah, blah, blah. In your notes on day 2, there is a misprint, Katie. I'm pretty sure it says DNA polymerase 3, but it should be DNA polymerase 1. So there's the enzyme DNA polymerase 1, comes back, it takes out that RNA primer and adds in the appropriate nucleotides that should go there, okay? Now, when all that's said and done, those Okazaki fragments still are not connected together. So there's still a gap. So number 7, Katie, what happens?
DNA ligase joins the...
Blah, blah, blah. Okay. So DNA ligase comes along. It's like the sewer of the bunch here, and it's going to stitch all of those DNA segments, the Okazaki fragments, it's going to stitch them together. It's basically it's going to help them bond together so that you have one continuous strand of DNA there. Okay? Any questions? And so you have to keep doing this all the way down the strand of DNA, this leading and lagging strand. It goes in both directions. Like I said, Mr. Bozeman and it's Anderson actually, but Bozeman and Crash Course both have really good videos showing this, but I think you need to have a little bit of understanding before you watch them or it's probably not going to make sense. So, yeah. All right, 326. As Mason said, you don't want mutations. So if you read your notes yesterday thoroughly, you noticed that DNA polymerase lays down something like 500 nucleotides per second. So imagine this enzyme that's throwing on 500 nucleotides per second. It's mind-blowing. I don't understand it. I can't believe it, but anyway. That's a lot of nucleotides per second. You would expect some mistakes to take place. That's like typing. You normally, you can type pretty good at like 50 words per minute, and then you're trying to do it at like 4,000, and you know, no telling what you've written down. But anyway, if there is a mistake, there's an app for that. I mean, there's an enzyme for that. So at the top of 326, it talks about nucleotide excision repair, basically where you're cutting out mistakes and you're replacing them. So that's in your notes at the end of part 2 notes, and it's also at the top of the page. Make sure you're familiar with that. Now, let us move on to protein synthesis, page 337. So not only is replication super important concept that you need to be familiar with all those enzymes and the processes, protein synthesis is also a very, very important concept that's complicated, that you need to be familiar with all the process and some of the enzymes, and we'll go through the different parts here. Now, your body is made up probably 50% protein. A lot of that structurally, and you also need proteins for most of your proteins are used to make enzymes. Good. What do enzymes do? They help to basically facilitate all the reactions in your body. Okay? So without proteins, you got a problem. Y'all probably remember from Lorenzo's oil. Y'all watch that? We're not going to have time in here. But Lorenzo had a mutation where he did not make an enzyme that helped to break down a fat, and the fat built up in his brain tissue, and it stripped away the myelin off the neurons, and then it caused brain problems, right? It causes neurons to not function. So, and I don't know how many mutations cause that, but I imagine it's probably a small number. I'll give you another example. Sickle cell. Y'all familiar with sickle cell disease? People have sickle-shaped blood cells. That's caused by a mutation in one letter. It's the 6th amino acid is switched because, it's the 6th, what you call it? Nucleotide, sorry. The 6th nucleotide is switched from like a T to a G or something. I can't remember which one it is exactly. But one nucleotide, one letter, like A, T, C, and G, one letter is changed, and it causes a complete deformity in the way that blood cells are made. So all of this is very important that it's done correctly, okay? So proteins are needed. We need them to make who we are, and we also need them to make enzymes and how we function. Where are we? Proteins are constructed from the macromolecule. Proteins are constructed from, So the monomers of proteins are amino acids. So if your protein is your train, the cars on the train are going to be amino acids. Now, there's primarily how many amino acids found in nature? You might remember in the beginning of the year? All organisms on Earth use the same basic 20 amino acids. Us, bacteria, viruses, whatever, okay? Amino acids are hooked together via what? Covalent bond. What do we call that specific type of bond? Number 2 on protein synthesis 1 notes. Read that for us, Mason.
Amino acids are linked by a covalent peptide bond by hydrolysis reaction.
Okay, there is something wrong in that sentence. What is it?
Hydrolysis.
What should it be?
Dehydration synthesis.
Very good. Hydrolysis does what? Break stuff. If you're linking amino acids together, making a protein, then you don't need to break something. You need to put stuff together. So you would use dehydration synthesis or what they call the other condensation reaction. So, yeah, that's a misprint there. Number 3, Mariah.
DNA is more stable than RNA, but RNA evolved before DNA did.
Okay. DNA is super stable. We have cells in our bodies, like brain cells and muscle cells, that the DNA is there in the nucleus for the duration of our lives. And it's never replicated. It's the same DNA you start out with, okay? RNA, it's a copy of it, just like if you have, this template, you make a copy of it, and it's like a piece of paper, and eventually it falls apart and breaks down. So RNA is kind of that cheap disposable copy, okay? So these two guys, Beadle and Tatum in 34, they developed this hypothesis that said one gene creates one enzyme. And so really we can kind of back this up a little bit. That is true, but it was changed to later on to one gene creates one polypeptide, or one gene creates one. A polypeptide is another name for protein. And then some of those proteins are used as enzymes. So here's the question we need to ask ourselves, Mason. What is a gene? You heard of a gene before? What is a gene?
Section of DNA.
A what?
Section of like DNA.
A piece of DNA that does what?
Like is the code for like some function in your body or something.
Yes. So a gene is a section or piece of DNA that codes for a specific protein. Okay? Now, like I said yesterday, and y'all weren't here, DNA in prokaryotes, like bacteria, almost all of their circular DNA codes for something. Like it's coding for something, some type of protein, almost all the way around. Our DNA there's a bunch of crap in there that's not needed, and it's been in there forever, and it's not very efficient in terms of the amount of DNA. It's estimated that maybe like 2 to 4% of the DNA that we have actually codes for proteins. There's a bunch of pseudogenes that no longer code for stuff that probably did a long time ago. There's a lot of viral DNA that's embedded in our DNA that doesn't code for anything. Just like when you get the herpes, it embeds in your DNA and it's there for basically ever. So there's a lot of stuff in there that's not coding for anything, okay? And that's going to be a problem we're going to have to deal with here in a second. Transcription. So before we get started with transcription, protein synthesis means what? To make. So protein synthesis, for you math nerds out there, protein synthesis equals what plus what? What two things make up protein synthesis?
RNA and
Good. Transcription plus translation. Okay? What do you think of when you think of that term transcription? What's a transcript? A copy of something. Like you might think of a scribe back in the day, someone who copied stuff before they had copiers. So transcription is just the process of making a copy. Translation is actually taking that information and putting it into some useful form. Okay? So these are the two components of protein synthesis. Transcription. Part A, making a copy of the DNA. Now, when we copy a gene, a segment of DNA that codes for a protein, Mason, what do we copy it into? What form? Do we need it to last forever? Do we need it to be super cool and stay around for the duration of your lives? No. So what do we copy it into? RNA. So we make a cheap disposable copy, specifically in this case, mRNA. This is going to be a message that's going to go out into the cell. Now, there are different types of RNA. We're not really worried about the other two right now. We'll talk about those later, okay? So, number one under transcription, Mariah.
The message mRNA will be sent to the construction site, ribosomes, for building the protein.
All right, so mRNA is copied from a segment of DNA. It goes out into the cell to a ribosome. What are the two ribosomes that it might go to? One on the rough ER, or it might be on the, just might be floating around the cytoplasm. If mRNA goes to the rough ER ribosome, it's probably making a protein that will go where? What? Nope. To the Golgi and then be modified and then leave the cell. The ribosomes that are in the cytoplasm, those are the ones generally making proteins for the particular cell. Okay? So, number two, Katie.
RNA nucleotides use ribose instead of deoxyribose as a 5-carbon sugar. This makes the RNA less stable than DNA. That is good since the cell only needs it to send a temporary message to construct the protein.
Okay, why does it only need to be temporary? Like, let's say that you go eat lunch here in a second, and your body sends a message to your stomach cells and says, "Hey, start making some enzymes to digest this food." Why do you not want that message to stay there forever?
Because you don't have food forever.
Yeah, so it needs to send out, make a message, make some enzymes, and then it needs to break down because you may not eat food for another 6 or 8 hours. So it's broken down, it's recycled, and that message can be used again. Okay. Number 3, remember RNA has a U for uracil instead of thymine. Excuse me. Thymine is not used in RNA. So the rules of base pairing still apply though. In part B there, A and U are always still going to go together, or they're going to be complement one another, and C and G are going to complement one another. It's just that U replaces the T in Chargaff's rule. Okay? All right, part C, Mason?
In eukaryotic cells, the mRNA must first be modified before translation can occur.
Ooh.
The modification occurs in the nucleus. Prokaryotes do not modify the mRNA. They send it to the ribosome as is.
Okay, so on page 337 on the right side here, at the top, the bacterial cell, a segment of DNA is copied through transcription, made into mRNA, and then that's sent directly to the ribosome where a polypeptide or protein is made. Okay? Now, in eukaryotes, what do you notice is different?
There's a nucleus.
Okay, there's a new. What about the process, though? The process of protein synthesis that's different.
It has to be processed before it can translate.
Okay, so it has to undergo some processing. The primary transcript, primary mean first, is not used at the ribosome. Now, why is that? So I'm going to give y'all a preview for later on in the notes. Turn over to 343. 343 in your book at the bottom. All right. Initially, from the DNA, you have this pre-mRNA that's made. The pre-mRNA and the mRNA, you'll notice a little bit different here. What do they put on the front and back side of this mRNA?
The cap and tail.
Why do you think it has a cap and a tail? Why do you think in eukaryotes they would put a cap and a tail on their mRNA? If you were sending an important message out somewhere and you were in charge of something, what would you might do to it?
Prevent that signal from going somewhere else.
Maybe. You're thinking too hard. You're too Star Warsy. It's not the Mandalorian here. So the cap and tail are simply just to prevent erosion away by enzymes that it may encounter on its way to the ribosome. Okay? So it's just for protection. So just like you might, if you were sending a memo to someone out, instead of just sending a piece of paper out in the mail, you're going to put it in an envelope and, something to protect it, to keep it from getting torn and messed up. And so that's where the cap and tail comes into play. Now, what else do you notice going from the pre-mRNA or that primary transcript to the secondary transcript, where the mRNA that actually goes to the ribosome? What is different there?
The intron is not there.
Yeah, so remember how I said there's a bunch of DNA that we don't use? So introns are DNA that is not coding for something. So introns have to be cut out, okay, using an enzyme, cut out. Exons are stitched together, and that forms that final coding segment. An easy way to remember this is exons are the ones that are exiting the nucleus, and introns are the ones that are staying in the nucleus, okay? Now, we talked about in the immune system, remember how I said that we have the ability to make almost any type of antibody? I don't know if y'all remember that. The reason for that is, imagine we have a bunch of these different exons along this primary transcript. When your cells, so your immune cells, when they go to splice these together, they can basically, I'm giving you the simple version here, they can splice them together in different orders. When you put those A, T, C's, and G's together in different orders, what's that going to do to your protein that results? It's going to change the shape, structure, which is going to change the function. So imagine an antibody, one maybe looks like this, and one looks like this, and one looks like this, and one looks like this. And so these antibody structures that you have, the ability to encounter the outside of like a virus, like the coronavirus, is going to be different. So the more, it allows you to create, I'm not going to say infinite number because it's not an infinite number, but millions of different types of antibody structures that you can, combinations that you can use to grab many different types of viral proteins, okay? And help to fight those off, or any type of really foreign agent that you encounter. All right, let's flip back here. 337, let's see. The primary transcript, that means the first copy of mRNA, has to be modified. Part D, this is considered the first part of protein synthesis. Excuse me. All right, part 3, translation. Translation is where the mRNA goes to the ribosome, and that mRNA segment is then going to be translated into a protein. So what does that look like? It's a good question. Turn over to 345. I'm going to give you the basic breakdown, and then we'll get in the details here. So it makes sense. All right, translation. Translating the message. Part A, Katie.
In this process, the cell is turning nucleotide language, DNA and RNA, into amino acid language to make proteins. Remember, amino acids are the building blocks of proteins.
So on page 345, let's just look at the picture real quick here. So what's the big, at the top right, that 1714, what's the big brown blob? What is that? That's the ribosome, okay? So that's where proteins are made. What is the long reddish orange strand thing at the bottom? That's the mRNA. So it has different segments called, and you're going to see this codons. Those are the different codes that code for specific amino acids, okay? So the mRNA has different sections that code for specific amino acids to give you a specific type of protein. What are the things that look like little green inhalers? T stands for?
Transfer.
Yes, these are the transfer RNA. They transfer amino acids to the ribosome. Why do you need amino acids?
Make proteins.
To make protein. Good. So, and they have to match up with a specific codon in order to be able to drop off that amino acid. What is the big chain of purple things that looks like a kite floating around?
Protein.
That's the protein or polypeptide. So, and they're hooked together there at the ribosome, and it just keeps adding them on as they come in, okay? Until you get to whatever your primary protein structure is. Then remember all that stuff we learned about. It has to go fold, it has the hydrogen bonds, the primary structure, the secondary, well, that's this primary, the secondary structure, the tertiary structure, and quaternary structure. So all that stuff has to happen afterward, but this is how the polypeptide is put together initially, okay? All right, let's stop right there.